Okay, so I think we should get started. Hello, everyone, and welcome. Thank you for joining today's insightful webinar. I am your host. My name is Monica. I'm the head of go-to-market at My Laboratories. So first, I'll start with some housekeeping items. Feel free to drop any questions in the chat throughout the session, and we will dedicate some time at the end for Q&A. The session is being recorded, and of course, we'll share this and additional resources afterwards. So moving forward to the topic, you've probably wondered what really happens to my immune system after a vaccine. And so today we'll answer exactly that question and use multiomics data to support the conclusion. For today's agenda, we have Khadija, who is a product manager at Cellecta, and she will lead us through the vaccine case study, the study design, and the technology behind the assays. And then Stan, the CEO of My Laboratories, will talk about bridging the gap between the raw data generated from that study and how we can obtain actionable insights. So I'm sure you guys are all extremely excited. So let me pass it over to Khadija to start.
Thank you, Monica, for hosting Cellecta at this webinar. I will be presenting the study that Cellecta has done regarding vaccination. And before we get started, let me introduce you a little bit about what the technology is and the benefits of using this technology. So, adaptive immune receptor profiling is done to your T cells or B cells, which are part of your immune response. So, as everyone knows, when a pathogen attacks your body, there is a certain type of immune response that your body exhibits. The first line of immune response is your innate immunity, which happens immediately. It can come off as an allergic reaction, hives, sneezing, and then and then it triggers another immune response, which is the adaptive immune response, which is also called a delayed immune response, which takes a few days to kick in. And this session will be focused on adaptive immunity, which is primarily regulated by two types of immune receptors, which is T cell receptors and B cell receptors, which are on top of your T cells and B cells. So, what are some advantages of studying this type of technology and how it can help in your in your research? So, um, one of the primary reasons for doing biomarker discovery, we find genes that are upregulated or downregulated by a certain type of pathways or responses to drug treatment. In this one, it's not just your genes that are regulated, upregulated or downregulated. You also get to understand the dynamics behind those transcriptomics. So, for example, you can understand the immune responses or the receptors that are responsible for that type of gene upregulation or downregulation. So, the type of studies that can be done with this research is you can do a predictive or prognostic biomarker discovery, or you can do therapeutic targets based on a certain type of drugs, like immunotherapy that could be delivered to cancer patients. You could also do personalized immunotherapies. And then the most common usage of this type of technology is in blood lymphomas, where the patient is monitored throughout the course of the immunotherapy for tracking how many T cells or B cells are upregulated or downregulated during the course of the treatment. Just a little bit about the biology of this type of response. So, as I mentioned, the T cell and B cell receptors, which are top of the TCR and the BCR, have a unique mechanism, which is called the VDJ recombination. And this allows them to have the type of diversity that is required to exhibit an immune response. And to give you a reference, on general, we have about 19,000 protein coding genes, whereas the diversity of these TCR and BCR are in hundreds of millions of them. And the reason is that our body gets attacked with multiple different types of pathogens throughout our life. And in order to be able to sustain or react, we need to have a lot of diversity to be able to recognize each and each and every different type of it. So the way these receptors are formed is that the genes that encode them have the V, D, J, and C segment, and in order to exhibit diversity, it translates into taking one of each of these portions, so V, D, J, and C, and hence you can get multiple millions of recombination events happening in this, which causes it to have a very diverse immune response. So, just to understand this a little bit from some studies perspective, so this was a study that was conducted on 5 individuals to study their immune receptor profiling from genomic DNA and mRNA. And the goal of the study was to compare the similarities between individuals and also to see how similar are immune receptors on the genomic DNA versus the mRNA level. And on this plot on the right-hand side, you can see this is genomic DNA, and on the x-axis and on the y-axis is mRNA. And you can see that there are certain clonotypes. Um, Let me just use my pointer. Uh, there are certain clonotypes that are only present on the mRNA level, on the transcript level, which are not present on the genomic DNA level. Whereas there are certain genomic sequences that are only present at the genomic DNA level, which are difficult to capture on the RNA level. And when they studied that, what is the shared clonotypes, and they were only able to find 13 to 40%, 14% of shared clonotypes between genomic DNA and RNA, and rest all others of these are unique to mRNA and genomic DNA. And this is an interesting factor in this type of research. And this kind of was the impetus for Cellecta to come up with a technology that that not only focuses on genomic DNA and RNA, but both on the RNA and DNA level. So, to explain what happens on the biology side, when an antigen is recognized by a naive T or B cells, it sends it a signal to proliferate. So the number of cells actually increase. So as you can see from one cell, it becomes two cell over here. And then at the same time, on the RNA level, the transcripts also increase. So from 3 transcripts from RNA, you can see multiple transcripts here. So in terms of the fold change increase, we're actually seeing from the naive state to the activated state, the genomic DNA, you can see around 5 to 10-fold more cells, and each cell has one unique DNA. But on the transcript level, you can see anywhere from 50 to 1,000-fold activation, depending on what type of genes they are. And this is something that was intriguing for us, and that's why we wanted to have a capability that can provide insights from both DNA and RNA level. So just to explain you a little bit about the driver map technology. So this is a multiplex RT-PCR assay that can be done in a single day. It has PCR steps that can be conducted by any lab technician or scientist. And then the following is an NGS profiling step. And it can the way that our assay is designed is that it can cover the CDR3 or the full length amplicon region, which covers the CDR1, 2, and 3 regions. And we have validated this with multiple redundant V primer sets. So it helps that to really make sure that all the genes are captured when you're doing immune sequencing. And then it has anchor primers that are used for unbiased amplification. And then the way this technology is designed and optimized is it really helps with avoiding primer dimers and off-target products. So it's very specific to the type of amplicon that you would like to amplify. And then, in conjunction with that, to make this analysis more quantitative, we have included validation barcodes, which is Selecta's proprietary technology, which in short stands for VBC. And in the upcoming slides, I will explain more how this VBC can actually make your analysis even more quantitative. So just to explain you a little bit about the design of the assay. So in terms of RNA, we have two options. So your first option is to only sequence the CDR3 region. So the primers bind to the FR3 region and the reverse primer to the C region. So this is about a 300 base pair amplicon. The second option is to sequence the full length receptor region, which is all the way from FR1, CDR1, 2, 3, all the way to the C region. This is around a 600 base pair amplicon size. So it really depends on the type of studies that you're doing, where one is better than the other. But most commonly, the CDR3 is the region that contains the most diversity. So that is of interest to a lot of people. And then on the genomic DNA side, the reverse primer binds to the J region and forward to the FR3 region. And hence you can see that if you sequence both the RNA and the DNA simultaneously, you can see the overlap of CDR3 region between both of these two methodologies. So as I was talking about, the benefits of using Cellecta's assay is that we have a flexible input. So you can start your study with RNA or DNA. And it actually really depends on what type of samples that you're using. For certain samples, such as FFPE, tumor biopsies, where the RNA is degraded, the DNA is a good choice. Whereas for other samples where there are enough starting material, such as PBMC, whole blood, we recommend RNA. And if you want to go a step further, you can use both DNA and RNA to figure out the activated clonotypes. The other advantage of our technology is using a cell direct protocol. So for samples where the number of samples is very limited, number of cells is very limited, you can directly profile from number of cells without having to do any RNA extraction or enrichment process. And additionally, we also have add-on protocols for genome-wide expression profiling and single-cell TCR BCR chain pairing or immunophenotyping assays that you can add on to get further insights. So, to talk a little bit about the capabilities of our assay, as I mentioned, you can do both RNA and DNA. And on top of that, we offer an assay that is very comprehensive, that you can profile all TCR alpha, beta, gamma, and delta chains, as well as BCR, HKL chains in a single reaction for RNA and two reactions for DNA. And the reason why we wanted to come with an assay that is very comprehensive is that there are certain studies that show that TCR alpha is associated with certain type of cancers, or gamma delta chains are affiliated with certain immunotherapy response. So we wanted to provide a comprehensive solution to our customers so they can get all of this information in one single reaction. To talk a little bit about the sensitivity of our assay and how we are different from other competitors on the market. So this figure on my left compares TCR clonotypes from driver map air assay to TCR clonotypes from smart technology. And you can see that from alpha and beta chains, we can detect almost 3 times more sequences as compared to smart technology. And this is also consistent with immunoseq assay, where you can detect around 1.5 to 2 times more sequences from starting with driver map air RNA as compared to genomic DNA-based immunoseq assay. So, talking about the quantitation side of the assay, I mentioned that Cellecta has a proprietary validation barcode technology, which stands for VBC. And what it is that it is a set of 8 defined barcodes, which are 6 nucleotides each. And they work as internal replicates for quantitation, error detection, or sequence correction purposes. And what is different about this is that instead of short defined having random UMIs for quantitation, we have come up with our own technology with 8 defined barcodes, and it's really helpful in terms of um, sequencing the same amplicon with 8 different barcodes. So you can collapse the reads together and get a more robust idea that if this amplicon is actually true or not. So it really helps with having more confidence in the data analysis side. And then it also adds more specificity because it does not have primer dimers as it can have in a UMI-based assay. So we did a study to compare BBC technology with a UMI-based assay, which was our driver map one version 1 assay. So we were able to see that from lane 5 and 7, this is for VBC for both TCR and BCR DNA, we were able to get much stronger bands for consistently for both TCR and BCR as compared to UMI-based assay, which is 6 and 8. And moreover, when we did the data analysis and we dug deep into this, we were able to find on the right on the x-axis is VBC, VBC based for 50 nanograms, and then on the y-axis is UMIs for the same 50 nanograms. And overall, it has a linear correlation. So most of the clonotypes are overlapped, but there are certain clonotypes that you can only detect in the VBC-based assay for both BCR, IgH chain, and for the TCR, TRB chains. So it shows that there are certain benefits of using VBC technologies versus UMI in terms of sensitivity and specificity for this type of technology. So now, moving forward into a vaccination study that we conducted with actually one of the volunteers at Cellecta. The study was designed in a way um, that just to give you a little bit information about the profile. So this is a male 70-year-old participant who happens to be our CEO, and he was given 2 shots of vaccinations. One is the tetanus and pneumonia. Um, and the second is Shingrix vaccine. And we took blood samples before the vaccination event happened, at the day of vaccination event, and over a period of a couple of days. And then after a few months, to see how the immune response has increased and then come back to its normal level. So we had controls from before the experiment as well as after the experiment. And we did 3 different types of analysis on this sample using driver map technology. So the first was a gene expression profiling assay for sequencing 19,000 protein coding genes. The second one was single cell RNA-seq. And the third was TCR-BCR immune receptor profiling. I'll show you results from driver map expression profiling assay. So over here, you can see that at day 2 post-vaccination, there are certain genes that are upregulated at day number 2. And when you see these pathways, what are the pathways that are associated with these genes? We were able to find them associated with innate immune response, which is the frontline immune response. Versus day 8, the genes that were upregulated were associated with adaptive immune response, which is the later immune response that occurs around day 7, day 8. So this gave us confidence that our results from from expression profiling from vaccination study have a correlation between innate and adaptive immunity. Further on, we did immune receptor profiling analysis from both DNA and RNA. On the left-hand side, you can see TCR beta chains from DNA and TCR beta chains from RNA. And on the right-hand side, this is IgH, which is B cell receptors from DNA and RNA. So starting with B cell receptors, on the RNA level, you can see that a lot of genes are upregulated. But whereas on the DNA level, there are fewer genes that you can find, or clonotypes that you can find that are upregulated. And this was interesting for us to see. That comparing both RNA and DNA results, you can actually figure out what are the activated clonotypes that could be overlapped in both RNA and the DNA level. And same response on the TCR beta side, the response is not as strong as B cell receptors, but you can see similar trend where RNA genes are much upregulated, whereas DNA you're finding much lower clonotypes. So we wanted to dig deeper into this. So we asked our collaborator at Platforma to help us with this analysis because, as you can see, this is quite complicated. And I would like to now pass on to Stan to do a deep dive into immune receptor profiling data and show you some insights that he was able to find for us.
Thank you very much, Khadija. Really interesting. Thank you for this deep dive into the wet lab part of things. Now I'm switching to a practical part. I'll just share my screen and go through some analysis to show how you can use this data to get insights yourself with our software. Yeah, let me just share my screen. And you need to stop sharing before I can start sharing. Yeah. So that's it. You see my screen? Yeah. Very well. So yeah, let's go straight to the point. Here you see one of the final results that I wanted to show during this practical session. It's actually a clonotype space, a space of all B cell receptors with some of them highlighted. Those are specific B cell receptors that were activated during the first round, the first immunization campaign. And this is the whole space of all B cell receptors that we have in this data. As you see, it is a big thing. And this is one of the final results that I'm going to show you during this practical session, the clonotype space and how we can find something interesting here. For example, those B cells that were activated during the immunization. So you see the Platforma software right here. It's opened. It's basically a graphical user interface. It's a desktop application which you can download, by the way, from the Platforma bio downloads. It's absolutely free for academic research, and you download it, install it for your Windows, Mac, etc. And there is a desktop application which you can use to run all the analysis. At the same time, you can connect it to a server to offload some heavy calculations. And let me start by going from the very beginning. Let's say when you do the analysis, you start from the new project. Let's call this project, my project demo. And the first thing you're going to do, you're going to import your sequencing data. That's the data that we discussing on Khadija's slides. Yeah, Platforma is actually a platform where you have different applications for different types of biological analysis to get different types of insights for different biology areas. And as you see, the first thing I'm doing, I'm clicking Add Block, and in the block registry and the block gallery have many different applications for various types of the analysis, including various multiomics applications. So we have support for gene expression profiling, single cell, and of course VDG analysis with MyXR, the tool that we developed. At the first point, the starting point of any project is samples and data. There's the place where you define your project and your samples and import the FASTQ files. There are two options. You can import your FASTQ files right from your computer if you run Platforma locally and you want to perform all the analysis on your PC, or if you have connect Platforma to a backend to your, for example, institutional HPC cluster or just to your cloud server, then you can also connect to remote and import FASTQ files from the remote storage. So for this demo, let me go and import some FASTQ files that I have for the vaccination project, for the vaccination study that Khadija presented right here. So I have FASTQ files, and this app, this block, samples and data will automatically infer the structure, the file name, read one, read two. If you have lanes, the lane structure will be also inferred automatically. If you have, let's say, plate-based protocols or something else encoded in the file names, it's all possible to adapt here and ingest this information as a metadata. So let's call this data set RNA. This is a RNA data set, RNA air. And we have our data set with all our samples and the FASTQ files, pairs of FASTQ files for all of our samples. In the same way, we can import multiple data sets. We can import the DNA data set, the gene expression data set, the single cell data set, the VDGA and gene expression part, whatever, and then do the analysis, the downstream analysis on all the data sets and integrate this data together. That's one of the key features of Platforma. But for the sake of this short webinar, I'm going to focus on the VDGA analysis of the RNA assay. So we have our samples, and then we can import metadata corresponding and defining our experimental setup. So if you have an Excel file or TSV file with whatever information about your samples, you can import them, and this will be the universal, let's say, entry point to any project. You have the data sets, and for every sample, we define samples. For samples, we have FASTQ files and various metadata. And this metadata is actually absolutely arbitrary. You can have whatever metadata columns you have in your study. In this specific study, our key metadata is time point. This is one patient, the president of Cellecta, and the time points for the first vaccination and the second vaccination. So once we click run, this will import all the FASTQ files from the storage into the project, into the Platforma. And our next step, and the obvious step for this specific VDG analysis, is to get clonotypes from the FASTQ files. We work with Cellecta for quite some time, and Cellecta has their own block. It's called driver map air clonotyping block, available in Platforma, which actually under the hood uses MyXCR tool to process all of the upstream files. We select the data set that we want to analyze. Here we have a type of chemistry for Cellecta. So all of the types of kits that Cellecta offers, we have here. You just pick the name of the chemistry. In this case is driver map adaptive immune receptor human RNA assay, and you click run button, and then the analysis will start. I'm not clicking run button right now. This is a huge project. There's 200 gigabytes of data, and I don't want to run it on my computer right now. I have already processed data. I have already processed a project where I processed all those samples before our webinar. So when you click run button, the analysis will start. If you have a backend configured for Platforma, then the analysis, this 200 gigabyte processing will happen on the backend, obviously. And here you will see the progress. And the analysis here in Cellecta driver map block is tailored specifically for the Cellecta chemistry. So the presets that you have here for the Cellecta chemistry, they use MyXCR under the hood, and all the parameters are specifically optimized for this chemistry. So you will have the quality controls. If you click on the individual sample, this is very useful because here you can see the overall, you can go deeper into individual sample and see like whether everything is good in the wet lab. Like the alignment rate is pretty good here. Also, one of the nice features actually of this specific assay is that you can profile all of the genes simultaneously, and that's what we have here. So here we see the distribution of the immunological chains in our sample, and we see, quite even distribution. It's actually, very nice. Although we see that like light chain is slightly more, and general antibodies occupy more space in our sample, which is something we expect, actually. It depends on sample, of course. We can go to the quality checks to check, to read whether everything is good in the wet lab, or there are some alerts or warnings, something that typically provides you a ready-to-use feedback to the wet lab. What potentially may be improved, or if you have alerts, then what should be fixed in the wet lab regarding the quality controls. And all these quality controls, they are specifically designed, specifically optimized and tuned for the type of chemistry is used here. So yeah, that's the upstream part of the analysis. And now let's go and try to get some insights from this data. So the first thing we want to do, we have the clonotypes. We want to look on the clonotypes. Just, well, the first thing any person want to do, just look. Yeah. For that, we have a clonotype browser. That's a nice app allowing to just look at the clonotype tables. Yeah. Here we select the data set. Let's say IG heavy. This is quite large data set. So we have a table of the clonotypes, all the clonotypes. Here we can switch between samples. There are many columns available with all the information about like CDR3 sequence. This is a CDR3 sequence and only. So we have the information about the CDR3 sequence, nucleotide, amino acid, protein sequence, genes, the abundance of the clonotype, and all of the properties that you typically expect to have for this type of data. If we go to the overlap, then in the overlap, we will see all of the clonotypes from all of the samples in one table. So specifically here for this data that I present for this webinar, we have 200,000 of here we selected immunoglobulins, B cell receptors, 200,000 of B cell receptors in this table. And here, actually, you see, let's say if I sort by the number of samples, you will see that some of the B cell receptors present in all of the samples that we have. Well, that's obvious because this is one patient. Yeah, so it's good that we have this. Yeah, and also you can track the abundance on the individual clonotype across different samples. This is boring, but this is what the first thing everybody wants to look. And now more interesting stuff. So table with 200,000 of lines is not what people want, and I don't like it. Well, of course we need it, but then let's have like a bird eye view on whole space of our antibodies or B cell or just B cell receptors. And for that, we have a nice application, a nice block. It's called Clonotype Space, which actually performs a dimensional reduction of the clonotypes based on their sequences and projects them on the UMAP plot. So as you see in the settings, I selected the B cell receptor from the data. So I'm looking only on the B cell receptors, heavy. Yeah, obviously for single cell data, you can look on the cells here, but for bulk sequencing only it. There are some parameters. We are not going to look at those advanced parameters with the defaults. We're already good to go. And here we see the space. The idea of the space is that two clonotypes will be located closer to each other if they share similar properties. And those properties are defined and determined by the protein sequence of the antibody. And we see some nice clustering, some nice grouping here, which is interesting. Yeah, we will look on this further. What we can do, we can also use a lasso tool to select a group of our antibodies and look on them with a multiple sequence alignment to see how they different or not. Like in this specific group that I selected, there is one outlier. Yeah, but all others, they are pretty similar. So yeah, and yeah, that's good. What we can do next to just understand what we have here in our data, we can, for example, color and group our clonotypes by, let's say, best V gene. Let's move the best V gene, which is a metadata in the graph maker. That's a generic instrument we have in Platforma, graph builder tool, as a grouping. Yeah, to color our clonotypes by the V gene. And yeah, and we see actually some structures. We see some structures. So this is a nice tool to have a bird eye view on our clonotype space. Let's use this tool to find something interesting. How to find something interesting? There are applications, there are blocks for this in Platforma. Well, let's start with something simple. Let's say V gene usage. This is a block allowing to calculate the gene usage in our samples. clonotypes and samples. So again, let's focus on the heavy chain, for example. Let's run it. It will run some calculation to calculate the gene usage. Done, calculated. And here, for example, we see the V-gene usage. If we zoom in, we see our samples. Yeah, if we zoom in more and more, well, that's too many genes. If we zoom, then we will see even V genes here. Yeah, but that's not super important. Yeah, and we see some structure, we see some bias, we see some really overrepresented genes. Also, we see some clustering here. So we see some samples, they group together, some genes are grouped together. This is interesting. Yeah, something is there. How we can focus on this, how we can go deeper. Again, let's say we have this graph maker. Let's bring something to visualization. Let's say we have a time point. That's a key metadata that we have in our project. We are doing a longitudinal analysis, and the key direction of our investigation. And let's use time point as an annotation layer to see whether the grouping of our samples based on the abundance of certain genes correlate with the time points. And yes, we see nice correlations. This group, this group, this group. So we see something. So it's definitely something interesting. Let's go further. Let's take some gene, some specific gene. This one, IGHV315. It's interesting. Maybe this is a nice gene. And because it's really very abundant in all of the samples actually. And let's go and look how this gene, how clonotypes sharing this gene located on our clonotype space. And how to do this. For example, like this. Let's take the best V gene and go to the highlight. And here I am copy pasting this specific gene. Okay, so you see how those clonotypes present and located on our clonotype space. What can we do else? Let's use J gene as a coloring. Well, we see something nice. We see that actually the big clusters on our clonotype space correspond to the J gene, and the most abundant clonotype is actually well represented in all of the clusters and all of the different J gene combinations. Okay, returning to the fact that this is a longitudinal study and we have a time point, before going deeper and deeper, let's see, we know that this is a vaccination project. Since this is a vaccination, we expect there are two vaccines that Alex took. There are two vaccines, and we expect certain patterns in the diversity and in the shape of our repertoire at different time points if the immune response was actually activated by these immunization campaigns. The another obvious instrument allowing us to do this is a repertoire diversity. Let's say we add this block and run the diversity analysis again on heavy chain. Yeah, the same can be done for TCR beta or TCR alpha or whatever. Run with the default parameters, the diversity analysis to calculate the diversity of our heavy chain immunoglobulins across all of the samples. By the way, under the hood, this will calculate different diversity metrics like Shannon-Wiener, entropy, observed diversity with proper downsampling and normalization, because every time you take different amount of RNA, different number of cells, et cetera, so it has a proper normalization. So when it's done, we can go to the diversity graph and look at the diversity across different time points. For that, let's go here and let's remove sample from the primary grouping, and let's use time point as a primary grouping. Let's also go here and select different visualization. You see so many visualizations are possible. So there are actually so many ways how we can look on the data with this tool. And let's use line plus jitter dots. And also let's go here and order our points on the plot according to the actual timeline. Since in the metadata we have human like labels for the time points, it's not necessarily obvious how to sort them for the machine. So let's regroup them according to the actual timeline. So we have 2 days, 5 days, 8 days after immunization 1, 15 days, and 22 days for the first immunization. So control before immunization 1, 2 days after immunization 1, 5 days after immunization 1, 8, 15, and 22. And then for the second immunization, second vaccine, 3 days, 6 days, 17 days. Very nice. And here we are looking at the observed diversity. And actually, for what we want to see, it's better to use Gini index, which is also calculated in the repertoire diversity. There is a Gini index that we calculated because it will show us the higher values of the Gini index will correspond to the lower diversity of sample. And once we did this, we see that at the 8th and 5th day of after the first vaccine, there is a bump, there is a peak, meaning that at this time points, the diversity of the B-cell repertoire in samples was the lowest, meaning that some clonotypes actually expanded, which is something we expect for like how immune system works. And the same, but a less strong peak we have for the second immunization. So this already tells us something interesting. We have good result. Let's explore further. Let's do the following. Let's find those clonotypes, specifically those clonotypes which are expanded, which are involved in the immune response for the immunization. Let's find them, track them, look on them, and understand what are the properties of those B-cell receptors. How we can do this? Well, one way to do this, we can go to, for example, Clonotype Browser, and it's actually a very powerful. I said at the beginning that this is a boring tool, but this is a very powerful tool. Here we have filters, and we can filter by absolutely any column, any data that is present here in our clonotype space, in our clonotype table. We can also annotate certain groups of clonotypes, save those annotations, and then bring to our visualization. So it's all possible here, very powerful instrument. But for specific purpose of time tracking, we actually have a dedicated app, which is called Clonotype Enrichment. So Clonotype Enrichment is a block that allows to calculate, to find the clonotypes which show like increase in the abundance or in differential abundance, depending on whether we have replicas or not, across different conditions. So we can use this block to identify what are the clonotypes that are expanded in the response to the vaccine. But if we think deeper than actually what we know from the immunology, then the level of individual clonotypes is not necessarily enough to like look at the whole picture, because clonotype, even distinct clonotypes, but with a similar protein sequence, may be correlated and involved in the response together. So we can do a clustering of our B cells to find the clusters of clonotypes which are involved in the immune response. And let's do this first. Let's first use the clonotype clustering app that we have to build clusters of similar clonotypes. I'm selecting again heavy chain, CDR3 amino acid sequence. It's the sequence that will be used for clustering. There are two ways how we can consider the clonotypes similar or not, by exact match or by alignment with, for example, a protein blossom matrix, which will take into account the actual protein properties. And let's say we want to consider two clonotypes belonging to the same cluster if they have identity of 90% and the length difference not more than 95%. So once we run it, we will have it will calculate it will run the analysis or the clustering of the clonotypes. And yeah, now we have clusters of similar B cells. And as we see, there are certain clusters which are pretty large, like this one, 62 proteins here in this cluster. Yeah, some of them I can see have identical protein sequences, meaning that they have different nucleotide sequences. That's why they're here. Yeah, and the size distribution of our clusters tells us that actually majority of our data is not clustered, but that's fine. And now we can run the enrichment analysis on the clusters. Here in the options, we select the abundance that we are going to use as a measure of the presence and expansion, and we can calculate the enrichment on clusters. As a condition, we select the time point, and here we select the conditions, the time points that we are going to track, like the first day, the second day, etc., etc. I'm not going to run it. I have it already executed here, and I'm going to do the results because it takes time to calculate this. So once you run it, you will have basically the table, which is boring. But let's go here in the frequency line plot. You will see clearly that there are certain clusters which show a real expansion at day 5 and day 8. Yeah, so here we see that certain clusters of the clonotypes really expanded, which is very nice. It tells us that, yes, this is clear signal, there is immune response. So we can also look at different angles on those clusters of clonotypes. Yeah, but what most important that we identify them. Now we can look on them here, for example, or in the browser or in other apps. And once we calculate this enrichment in the clonotype space and the metadata and everywhere in Platforma, we have these results. Let's say under the hood, there is a database which knows everything which was calculated in the project and allows to integrate all these sources of the data together on the plot or in the table. Here we are working with the VDJ data, but you can imagine same applies to single cell and integration of VDJ, gene expression, etc. And here we will have enrichment, maximum cluster enrichment calculated in this block, which we can use as a filter. And let's say use a really high number, 8. 8, yeah. So now on our clonotype space, we have only those clonotypes which belong to the enriched clusters. Very nice. Let's do the following. Let's use the cluster ID, that's a cluster ID that we calculated in the clonotype clustering, to color our dots, to see whether those clonotypes belong to the same clusters. And indeed, we see that this set of clonotypes belong to the same cluster. And the final thing that we can do, actually, we can use, let's say, the frequency at day 8, let's say that's the peak as a size of our dots here, to see like what are those like really enriched clonotypes and how they present, how they are located in the clonotype space. And we see, for example, this group of clonotypes, which we can select again with lasso and deep dive into what it is on the multiple sequence alignment. Here, let's say we can add a best V gene at the label, yeah, to see whether they belong to the same V gene. And yeah, we see actually that majority of them come from the same V gene. So this is just an example of how deep you can go with some of the instruments available on Platforma using Cellecta chemistry. By the way, this is a very, very good demo project with actually 5 million of antibody and TCR sequences under the hood of this data set, allowing to get lots of insights, both in the vaccination, general immunology, and things like that. And my role was here just to highlight you and show you in a practical way how you can do this with the Platforma software and Cellecta chemistry. There are more apps and more things, and of course you can go deeper and deeper, but we are short on time, so I'm finishing.
Thank you, Stan. So now let's pass it over to Khadija so she can talk about the conclusions of the study.
Thank you so much, Stan. Definitely, this has been really helpful for us to dip deeper into this really complicated data set. So, real quickly, I want to go over the last slide regarding what all you can get from this type of information. So, Cellecta offers chemistries for both TCR BCR sequencing. We're soon launching our chemistry for single cell immune receptor profiling as well for chain pairing information as well as immunophenotyping. And then we have expression profiling kits for bulk RNA sequencing. And the power of Platforma is that you can analyze all of this data set under one hood with a very user-intuitive interface, as you just saw in the demo. And just to summarize our conclusion is that so basically, doing both RNA and DNA can help you find some activated clonotypes that are um, that that you can only find when you do both RNA and on the DNA level. There are certain genes that are highly expressed at innate immune response at day 2 and adaptive immune response at day 8 post-vaccination. And on top of this, there are other analysis tools that we did not have the time to go into this today, but there are immune watch detect algorithm that you can add on to the Platforma software to find the TCRs that are associated with these clonotypes. So that is particularly very interesting for epitope discovery type applications. Um, and and basically just wanted to say that from a scientist perspective, I don't know how to use command line version. So this is very useful for somebody like me to play around with my data results, try different types of visualizations to see what is a graphical interface or graph that is helpful to produce for publication purposes. And just want to end off with our call to action. Cellecta is currently offering a 50% trial discount for the 24 reaction kit. So if you want to purchase TCR or BCR RNA DNA kits, we would be happy to extend that to you. And we also offer discounts for bulk purchasing. So if you're part of a core lab or hospital service lab, we can definitely offer special discounts for that. And from the Platforma side, it is free for academic use. Anybody can use that on the academic side. And for industry, you can reach out to licensing at Malab for more information regarding license. And there is a trial period where you can get to try out a demo project and see how it is working for you. And take it on from there. So this is basically from our end. If I would like to open it for any questions regarding the chemistry or data analysis, and we would be happy to answer any of those.
Okay, great. Thank you so much, Khadija. So we did have a few questions. If you have any that have not been answered yet, feel free to drop it while I read off some of the questions, just so all of you can see the ones that were asked by our audience. So the first one is from Artem. He says, do you always have to start from FASTQ files? Can you process FASTQ using MyXCR and then import the clonotypes into Platforma and do the further analysis in Platforma? So Stan, I'm going to pass that question to you.
Absolutely. We have this feature in beta version, but it's not available in public. In 2-3 weeks, it will be released as a part of samples and data, and the special block to import any air data, not only processed with MyXCR, but if you want with other tools also, no problem, even from Cell Ranger.
Great. Mustafa asks, can I use these plots for publications? Is there an option to export the graphs? Publication sufficient?
Yeah, we have an export option in the graph maker. It will export the image, what you say, in an SVG, a vector format, which you can further convert to PDF or whatever format needed for the publication. And it will export exactly what you see. So you can play with the colors, you can play with the order, you can tune it to whatever you need, then export.
Okay, great. We have another question. With this program, is it possible to easily check the upstream and downstream nucleotide sequences flanking my CDR3 regions? If so, how user-friendly is this process?
This question goes to our Chief Technology Officer, Dmitry.
Yeah, sure. Sure, you can inspect the data that is covered by the protocol, depending on the data you have, like whole BDJ region or sometimes as narrow as CDR3. But if you have the sequences in your data, you can, in the clonotype browser, you can see them and inspect them. And in tools like clonotype space and in other tools as well, you can inspect multiple alignments of groups of clonotypes, selecting using different like clustering, selection, manual selection, filtering. And well, yeah, it is pretty much user-friendly.
And to add here, actually, for example, in this specific study, this vaccination study, the chemistry was CDR3 only, but Cellecta also has a full lens. So the same chemistry can be applied to sequence the full lens. Yeah, and you can actually do the same. You will see instead of CDR3, you will see all of the regions, CDR frameworks, etc., etc.
Okay, and a final question, also from Artem. Can I use the differential expression tool in Platforma for the analysis of differentially expressed clonotypes, or is it only for the transcriptome analysis? Dimitri, do you want to take this or Stan?
Yeah, let me answer because I already answered this in the Q&A chat. The differential expression is for expression data, and for clonotypes, there is a block that Stan showed during presentation called differential abundance. It can work either
Clonotype
on the
enrichment.
level of clonotype enrichment. Sorry, yeah, and it can work both on the raw clonotype level or on the clusters, depending on your task. If this is samples from the same patient, you do raw clonotypes, or cluster them if you have more diverse samples. Yeah, so for each type of data, there is a specific tool because algorithms are different, the specificities are different, the UI is different. And in addition to that, the output of this block can, if you do the campaigns of enrichments, then there is a seamless integration with downstream blocks where the lead selection blocks where you can integrate this information with sequence liabilities, the ML predictions of specificities of certain clonotypes to select like best sequences for future work.
Thank you, Dimitri. In the interest of time, we have one final question. This is coming from Mustafa. I think this is for you, Khadija. Can you explain the VBC technology and the chemistry behind it again? What is the advantage compared to UMI-based technology?
Yes, it's a really good question. So VBC is actually it's a set of 6 nucleotides that are actually I do have a picture if it helps explain, or we can probably look at the website. Is it okay if I open it up real quick?
Yeah, for sure.
Absolutely.
Okay. Just a second. Okay, so I think this image will explain it more better. So basically, these VBCs are a set of so these are 8 barcodes in number. Each barcode contains a set of 6 nucleotides, which are different, and it can amplify the same clonotype 8 times. And by 8 times I mean it's not exactly 8 times. It depends on the abundance of the clonotypes. But the idea is that each clonotypes will be amplified by 8 different barcodes. So when you are doing the data analysis step, you can consider them as one as one true as replicates of each another, right? Like when you're doing a technical replicate, so these can be considered as that, and you can in the data analysis you can just batch them all together and then use that batched information as a qualifier if this is a true clonotype or not a clonotype. And basically, this can over here with this example, I can probably explain it further. So if you have an abundant clonotype, that abundant clonotype will be picked up by each one of those VBCs. So VBC 1, 2, 3, 4, 5, 6, 7, 8. And each VBC will have its own number. So in this case, there are 8 VBCs, and each one will have their own frequency. And for the final data analysis, you collapse this all together, and this becomes there is an algorithm that Stan and our bioinformatic team has developed. So basically you batch it together and then it becomes as the final result for your clonotype for abundant clonotypes versus rare clonotypes. You would be amplifying that rare clonotype by only one VBC because it's present at much lower frequency. So it's much easier to identify it as a rare clonotype while using VBC technology. And I think if you have any other questions, probably Kit is our bioinformatician who would be also able to answer that, but we can also take that offline in the interest of time.
Great. Thank you so much, Khadija. And thank you to our wonderful audience. We love the questions. We love the curiosity. And just to wrap up here really quickly, we will be sending out the links. So the select information that Khadija shared, as well as how to get hands-on access to Platforma. We will be sharing it right after this call.
Thank you very